All right, thank you, Amit, for a wonderful presentation. So we're going to move on to our final speaker, Donald Phinney from the Scripps Institute in Florida, talk about more about MSCs.
Okay, thank you for attending this morning. I'm going to go in a bit of a different direction. So what I'm going to do today is present basically a data-driven talk, talking about mesenchymal stem cells. And it's basically data generated in our lab that's looking to try to unravel some of the complex biology of these cells. But I think when I go through the talk, hopefully you'll have a bit of a different perspective about how we typically look at these cells. And our goal is really to try to help improve donor selection processes and manufacturing processes to make MSC-based therapies more efficacious. So Dan gave a good introduction about MSCs. As he noted, these were originally described in bone marrow as hematopoiesis supporting cells that also have skeletogenic activity. And over the past 10 years, they've shown to have very potent anti-inflammatory and immunomodulatory effects. And so as you're probably aware, there's been a really inordinate number of clinical trials that have occurred using MSCs as the principal biological agent, well over 800 at this point in time. And these trials are directed at treating a wide array of conditions, including osteoarthritis, spinal cord injury, brain injury. But many of these are focused on immune and inflammatory ischemic diseases such as stroke, MI, graft versus host disease. And as we've heard from the previous speakers, some of these trials have been successful, so there's a lot of anecdotal reports of patients benefiting in early phase trials, but most of the phase 2 and phase 3 trials have yielded suboptimal outcomes or failed to meet their primary efficacy endpoints. And there's a number of reasons why predicting the outcome of MSC-based clinical trials is difficult, and I've tried to list some of those here. So the first is patient heterogeneity. So there's really no metrics that are available to stratify patients into responders and non-responders. And there's a lot of work being done in the field currently. One of the most long-standing issues is the fact that human MSC donor populations exhibit significant functional heterogeneity. And there's a lack of markers that relate phenotype to function of these cells. And this is compounded by the fact that GMP manufacturing schemes, we really don't know the effect of large-scale manufacturing on the composition of these populations and how it affects their potency. And in fact, a master cell bank of cells is really not equivalent to a batch of drug. The other issue, which further compounds these problems, is the cells are used for a wide clinical indications, and this is really interesting in that there's similar donor selection processes and manufacturing schemes are used to produce master cell banks to treat medically unrelated conditions. So the idea is that MSC is sort of a panacea and can be used to treat all kinds of conditions. And that really focuses in on the mechanism of action, which is largely indeterminate. It's thought that, in part, the host microenvironment drives MSC responses that are therapeutic, and that the stem progenitor functions are indispensable for their therapeutic use because everybody's trying to exploit their anti-inflammatory and immunomodulatory activities. So trying to address some of these limitations, there's a real push in the field to redefine MSC products based on their potency rather than their composition or matter or phenotype. And so a lot of groups are working on activity-based assays to read out potency in terms of angiogenic or immunomodulatory anti-inflammatory potential. There's a lot of matrix-based and multivariate analysis that are incorporating physical, transcriptomic, and proteomic properties of the cells to try to understand what donor populations are most potent for a given disease. We're very much interested in addressing that question, but we took somewhat of a different approach. So we used a system-based approach in my lab that was aimed at defining molecular mechanisms that confer interpopulation heterogeneity with the hope that this would reveal some intrinsic modulators of biological potency. And I'm going to share that data with you today. The reason we took that approach is when I was in the Center for Gene Therapy at Tulane University, we actually collected human MSC populations from over 300 different individual donors. And when you look at that many populations, you quickly realize that there's tremendous heterogeneity between donors. We were really interested in what drives that heterogeneity. So this is an example of 9 different randomly selected donors that we've studied in our lab. And you can see if you just read out growth rates by counting or viability by MTT assay, or look at their colony-forming unit fibroblast potential, which is a measure of progenitor frequency in populations, it's enormously variable across these donors. And if we looked at over 20 different surface markers, what we found is that there's no correlation between the expression of any of these markers with any of these differences in growth and CFUF activity. So in fact, most of these markers have very narrow expression range across all the donors. So through some genetics and genomics-based approaches, we actually focused in on a gene called Twist1, and we found that across these different donors, Twist1 levels vary quite significantly up to 6-fold. And Twist1 is interesting because it's a basic looped helix transcription factor that's actually essential for mesoderm specification in Drosophila and plays a big role in mesoderm fate determination and lineage specification during development. So it's an interesting gene based on what's known about its role in development. And so if we actually read out normalized twist expression levels across our 9 donors and plot it against cell growth and viability and CFUF activity, we found a very strong positive correlation. So we were intrigued by this, and we wanted to know if that relationship was spurious or causal. So we took a representative human MSC donor population, silenced twist using siRNAs, and found that in fact it significantly inhibited growth, reduced CFUF activity, had a minor effect on reducing viability, and slightly increased apoptosis, or the number of apoptotic cells in a population. So it looks like these correlations hold up because twist is actually driving growth and CFUF activity in these cells. So we know, as I mentioned, twist is important in lineage specification during development. So we went on to ask, well, does it have any role in regulating the trilineage potential of these cells? So in the bottom panel, we actually silenced twist expression. And what you notice is it doesn't induce spontaneous cellular differentiation. But when you expose cells to the appropriate inducing conditions, silencing of Twist1 significantly augments their ability to differentiate. And we track that back to the fact that when you knock down Twist1, you upregulate a number of master transcriptional regulators that drive fate determination to these lineages. And a number of years ago, we had done a lot of work on clonally derived human MSC populations. So we basically grew out over 100 clones and did clone splitting to look at their multipotency. And we went back and measured twist levels in some of these clones. And we found out that twist is most highly expressed in clones that have a trilineage potential and less highly expressed in clones that are restricted to bipotential and the parent population. So this suggests that twist is really a marker of a multipotent progenitor cell. We also were interested in what actually drives twist expression. So if twist is regulating growth and suppressing differentiation, it would be interesting to know what really is maintaining twist levels in cells. And for many years we had worked with FGF2 and we knew that this was a potent mitogen in MSCs. And so we just went back and looked at if we treat human cells in serum-free media with FGF, we strongly upregulate Twist1. That leads to an increase in protein accumulation in cells. If we go back and silence Twist1, we actually upregulate FGF2 and downregulate expression of FGF receptor 2,3C, which is the predominant FGF receptor 2 isoform in the cells. And conversely, if we silence FGF receptor 2,3C, we also downregulate Twist1. So this suggests that twist is maintained by an FGF receptor 2,3C axis in the cells. And so if we go back and look at FGF receptor 2,3C levels across our 9 donors and plot it against viability in CFUF, it in fact correlates. So there's a strong positive relationship between FGF receptor 2,3C levels and Twist1 in these cells. And if you silence FGF receptor 2,3C, you see similar effects to what you see with Twist1 silencing: reduced viability, reduced growth, reduced CFUF potential. So we were really intrigued by this and thought, okay, Twist is driving growth and survival and regulating cell differentiation. What else is it doing? So we did microarray, where we looked at gene expression changes in parental populations versus those where we transected cells with a scrambled or Twist1 siRNA. And we found about 550 genes that were downregulated and about 250 genes that were upregulated. And when we did gene set enrichment analysis, we found that most of the genes that are downregulated are involved in cell growth, cell survival, DNA replication, and many of those that were upregulated are involved in differentiation, and that's shown here in these network plots. So the downregulated genes map to RNA transcription, DNA repair, metabolism, mostly cell cycle and cell motility, and the genes that are upregulated map to ECM organization, TGF-beta signaling, integrin interactions, and also a lot of immune regulatory interactions, as well as p53. So Twist is known to actually bind to and regulate p53 activity. So this was all well and good, so we started to look through some of that data and ask, does Twist do other things that might be clinically relevant? And the first thing we noticed is that if we knock down Twist1 in MSCs, we downregulate a group of genes with angiogenic activity. So VEGF, CYR61, CCL2. And to validate some of this, we first looked at CCL2. We found a beautiful E-box in the CCL2 promoter, which is the consensus sequence that Twist binds to as a transcriptional regulator, and showed by chip analysis that Twist does actually bind to this promoter. And we did ELISA to show that if we actually knock down Twist1 or FGF receptor 2,3C, we can significantly reduce the amount of CCL2 secreted by these cells. But if we actually pre-treat the cells with FGF2, wash it out, then collect conditioned media and do the ELISA, FGF2 promotes CCL2 secretion. And we showed that this was through a Twist-dependent mechanism. So if we transfected the cells with a scrambled or Twist1 siRNA prior to FGF2 treatment, treated the cells with FGF2, and then read out CCL2 secretion, FGF2 was ineffective in the absence of Twist1 expression. So FGF2 drives CCL2 expression through Twist1 upregulation. Interestingly, we went on to show that Twist represses a number of proteins that have anti-inflammatory and immunomodulatory activity, which have been well described in the MSC field. So we were one of the first groups to show that MSCs secrete IL-1 receptor antagonists, which is an antagonist of IL-1. And so when we knock down Twist1, we see that IL-1RN levels go up, as well as TGS6, enzymes that are critical in the biosynthesis of prostaglandin E2, as well as heme oxygen 1. And if we do ELISA and read these out, we see that Twist silencing does induce secretion of IL-1 and PGE2. We also found that Twist silencing actually upregulates IDO1. So this is indoamine 2,3-dioxygenase. It's in the kynurenine pathway, and it's thought to be one of the major proteins that MSCs produce to modulate T cell proliferation. It's known to be potently stimulated by interferon gamma, so a lot of people pre-treat MSCs with interferon gamma to induce IDO1. And we found that if we do pre-treat cells with interferon gamma, we actually downregulate Twist1, and that leads to IDO1 induction. So to show, I didn't have it here, but we actually showed that Twist does bind to the IDO1 promoter, again at an E-box. And to confirm that, we actually overexpress Twist1 in a population and then treat it with interferon gamma. And cells that overexpress Twist1 are unable to induce IDO1 to the same level as mock transfected cells in response to interferon gamma. So Twist1 is suppressing IDO1 at the level of transcription, and interferon gamma treatment downregulates Twist to induce IDO1. So Twist is really tied up in regulating expression of a lot of these proteins that are known to modulate some of the anti-inflammatory and immunomodulatory effects of MSCs. So to confirm some of that molecular data, we did some cell-based assays, which are shown here. So looking at angiogenic activity, this left panel is culture of primary human aortic endothelial cells. And so for them to survive in culture, they need angiogenins like VEGF. In this assay, we just read out viability. So if you take conditioned media from native or scrambled transfective MSCs, It improves the viability of the cells significantly over no conditioned media controls. If you transfect the cells with a Twist1 siRNA or pre-treated with interferon gamma, they lose their capacities to support survival of these cells in culture. And if you treat them with FGF, you get an enhanced effect. We then went on to look at endothelial tube formation using HUVEC cells grown in growth factor depleted matrigel plugs. And the readout is similar. In this assay, growth media, replete growth media is the positive control. So you get lots of endothelial tubules formed. Serum-free media is the negative control. If you use conditioned media from a native MSC, you see enhanced tubule formation. If you knock down twist or pre-treat with interferon gamma, you reduce this back to the level of background. And if you pre-treat cells with FGF2, you can actually enhance tubule formation over what you see with conditioned media from native cells. Similarly, we've done a lot of work with MLRs looking at T-cell proliferation. An example of that is here. So in this assay, we treat peripheral blood mononuclear cells with anti-CD3 and CD28 antibodies. And we read out the proportion of Ki67 positive CD3 T cells by flow cytometry. And what you see is, again, a native population of MSCs shows the typical immunosuppressive effect, blocking T cell proliferation in response to receptor stimulation. If we go back and silence Twist1 or pre-treat those cells with interferon gamma, we can actually significantly reduce this ability even further. Dr. Patel talked about HLA2. When you pre-treat MSCs with interferon gamma, you're known to induce HLA2 expression. What's interesting is Twist doesn't do that, so you can pretty much get very potent silencing suppressive activity by knocking down Twist1 in these populations, but you don't see increased HLA2 expression. And the bottom panel is actually a cohort of several human MSC populations that we've obtained from different locations used in this assay. And what you can see is you actually get a very strong correlation with Twist levels based on the ability of the cells to suppress T cell activation in response to receptor stimulation. So it holds up across a number of different donors. So I've gone through some data fairly quickly and sort of cherry picked some of the more interesting things. A lot of this work was published a few years ago. The take-home message is illustrated here. So what's, I think, the two take-home messages is that the important thing is that the stem progenitor and effector functions of MSCs, so their ability to grow and proliferate, to differentiate, as well as secrete clinically relevant paracrine factors, is actually mechanistically linked via Twist1. And what's really interesting is these phenomena seem to be mutually exclusive. And because of that, you can actually specify them via a hierarchical process. So we actually think that Twist1 establishes an MSC hierarchy that predicts biological potency. So as shown here, cells that have very high levels of Twist1 are really stem progenitor cells because they hold lots of potential. They're competent to undergo trilineage differentiation. And they have a pro-angiogenic phenotype because they produce a number of angiogenins which are directly regulated by Twist1. As Twist1 levels drop, you actually upregulate master regulator genes that allow the cells to be competent to differentiate in response to the appropriate stimuli. But you also upregulate a number of proteins that have potent anti-inflammatory and immunomodulatory activity at the expense of the angiogenins. And I should also point out, I think it was on the slide, when you knock down Twist1, you actually induce some angiostatic factors as well. So the cells lose their angiogenic potential but gain this anti-inflammatory and immunomodulatory capacity. So we think that this can be very useful to predict cell potency under different experimental conditions. And so we tested this in vivo using our favorite mouse model, which obviously has limitations. So a number of years ago, we were one of the first groups to show that MSCs are potently protective in a mouse model of bleomycin-induced lung injury. And this is an example of some of that data that was published years ago. So in this model, this is a histological section of normal mouse lung that was given an intratracheal injection of saline. So you see the normal alveolar architecture. This is, I believe, 5 to 7 days following bleomycin, intratracheal instillation of bleomycin. So this induces a massive pneumonitis where most of the parenchyma is filled with inflammatory cells. And this is an image of a mouse that was given bleomycin and then given a venous injection of MSCs into the jugular vein immediately after bleomycin instillation. And you can see after about a week, the MSCs actually preserve most of the lung architecture. And you can read out metrics of lung inflammation. So for example, TNF, it holds true for IL-1 and IL-6. You give bleo, you get a very significant increase in TNF expression in the lung parenchyma. But if you give MSCs after bleo, this is greatly suppressed. And you can look at the scarring that occurs. So this level of inflammation causes pretty significant fibrosis in the lung after several weeks. And you can measure that with hydroxyproline levels. So again, bleo causes a significant amount of fibrosis, but the MSCs actually prevent fibrosis by blocking the acute inflammatory response. So we asked the question, do twist levels in different human MSC donor populations actually predict their efficacy in vivo using this mouse model? So we picked 2 donor populations that have very different levels of Twist1, which is exemplified by their differences in growth rate and CFUF activity, which are highly significantly different. And we put them in this model. And I'm showing you 2 readouts of this model. One of the metrics that we've begun to use over the years is body weight change, which is really a clinical score. So when you give mice bleomycin, they become very sick, they don't eat, and they lose a lot of weight. So this is, I believe, 7 to 10 days post-bleomycin exposure. You see the mice lose a very significant amount of weight and they're quite ill. And actually, if there's no treatment, most of these mice will succumb to their lung injury. If we actually repeat the studies that I showed you and inject a population of MSCs that are low twist expressors, 789L, right after we give bleo, it's actually quite protective. So a week out, the mice don't lose very much weight. And actually, if you observe these mice, they don't look any different than the saline-treated controls. They're quite healthy and happy. And if you look at bowel cell counts in their lung, there's very limited amount of inflammation in their lung tissue based on this readout. However, if we actually do the exact same experiment using a donor that has very high twist levels, 775L, it's actually not protective in this model. And in fact, in some cases, it seems to make the mice sicker. And again, there's no decrease in the amount of lung inflammation. So this is intriguing because in most studies, MSCs always work in mice, right? So we were very intrigued by this. We asked the question, can we take a good donor that is protective and actually make it non-protective? So we repeated the study with our low twist expressor, but we cultured those cells in FGF2 for a week, which we know induces Twist1 and basically promotes growth of the cells, which selects for twist high populations. And then we washed out the FGF and then treated the cells identically and repeated the experiment. And what you can see is that if the cells are actually expanded in FGF, then they're not protective anymore. The mice lose lots of weight. They have lots of immune cells in their bowel, and levels of inflammatory cytokines such as IL-6 aren't decreased. In fact, they may actually be elevated in these mice. So based on that, we've come up with what we're calling a clinical indications prediction scale. And we think that hopefully this will be helpful in trying to match donor populations and, more importantly, manufacturing schemes to specific disease indications. And we think what's useful about this is it's very simple. So this scale is based on Twist1 expression levels. And I've shown you that human MSC populations that express high twist levels, they grow rapidly, they're pretty robust, they make high CFUFs, and they're pro-angiogenic. And populations that have low twist levels don't grow well, they're not as robust, they don't secrete angiogenins, but they produce a number of anti-inflammatory and immunomodulatory factors. And what's interesting is twist levels are very strongly correlated with CFUF activity. So you can basically just take your donor, do a simple CFUF assay, and predict what the twist levels are, and then use that in the appropriate model. And I think this is advantageous because in the field there's been a lot of talk about we need some kind of standard MSC that we can base all our assays on. And I'm not a proponent of that because I have no idea where that standard is going to come from. You know, some people want to use an ES or an IPS-derived MSC. I don't think that makes any sense. So trying to standardize the field against a standard donor population is almost impossible simply because individual donor populations have massive levels of heterogeneity. So whoever picks what the donor is going to really skew the field for some time to come. The other value of the CLIP scale is that it's based on multiple biologics. So we're really looking at the fitness of cells, how they respond to culture adaptation, and what kind of media you use to expand them. So we think that this could be very helpful, again, particularly looking at manufacturing. And I have an example of that. So the CLIP scale isn't going to be useful if it's not consistent across different independent donor cohorts. So I showed you all the data with our original donor population. We've been slowly collecting new donors from different sources. So the red is one outside source and the green is another outside source. And if we look at twist levels and correlate them against CFUF, they again highly correlate across these donors. And this is true for growth and viability too. The interesting thing is these donors were obtained from a group that grow their cells in platelet lysate, which is a high octane media that really drives expansion of MSCs. And almost all of those populations have very high twist levels. And so we're actually, as part of the ISCT, we're conducting a survey of all academic GMP facilities that make MSCs, and it turns out at least 80% of those facilities all grow their cells in platelet lysate. So, you know, culture is a selection process, and there's a lot of interesting data that's coming out. So people have done barcoding of MSCs, and they've shown that as you expand cells, you do clonal selection. I mean, that's obvious. So how you grow your cells is really important, and I told you in the beginning that there's really most of the clinical trials that have been done, there's no real selection of manufacturing processes. People just want lots of cells because they need lots of cells to treat lots of patients. So they've slowly evolved to this point where they can use media formulations that drive massive expansion of their populations. So they're really selecting for twist-high cells, which may be somewhat beneficial in cardiac trials. And I think answers why some of those trials, most of those trials show some metrics that show improvement, but maybe not so good in GVDH trials. And the data actually fit with some of those trials. So Katerina LeBlanc, who's done a lot of work with GVDH, she typically uses populations that are minimally expanded and plated at higher densities, which would basically select for twist-low cells. Whereas other groups who have done some of the phase 2 trials, they've done low-density plating as a selection step, which is going to select for fast-growing cells that have high twist levels. This is sort of an aside, but we've been trying to understand why are there intrinsic differences in twist levels between donors. And we know from old literature where twist was being looked at as an oncogene that the twist promoter can be heavily methylated. So my pointer is dying, but so basically the whole proximal promoter and even upstream to about -1 kb of twist is very GC rich. And most people have said that there's a methylated CpG island between -300 and the transcriptional start site. We looked across 1,200 base pairs of the proximal promoter, and what we did is we found an island that's out here that's highly CG rich and has a number of potential methylation sites. And so we actually took 2 new donors, one that's twist high and one that's twist low, again showing differences in their activity. And we looked at the methylation status of the promoter, and what we find is the twist low donor has a very heavily methylated island upstream in the Twist1 promoter. So we think as a start that probably methylation of the Twist1 promoter is driving some of these intrinsic differences between donors. We have no idea how this is actually regulated. And so this is something that we're exploring currently. So that's my last slide. I think we're on time. So in the end, there's been a lot of talk about, in the last decade, about MSCs as medicinal signaling cells, that you infuse these cells into patients and somehow they respond to the microenvironment and all is good. But the reality is the clinical outcome data aren't really very beneficial to patients. And so what we're basically saying is that you need sort of a stem cell-centric viewpoint when you approach clinical trials with MSCs. So you really should think about the cell as a whole, not just the fact that it can secrete some anti-inflammatory and some immunomodulatory proteins. But actually, all of these things are interconnected. And what's quite unique about MSCs is Twist seems to play a role in regulating a lot of these functions. So your donor selection, your media formulations, your criteria for expansion are all affecting the total cell and what its biology is going to be at the end point. And so I think this approach probably, hopefully provides a better foundation to develop metrics to predict interpopulation differences in potency and then to match donors and manufacturing schemes to disease indications. And so one thing we're doing now is to try to validate that is we're collecting MSCs, human MSCs that were used in different clinical trials for different indications and going back and applying the clip scale to them to see if they're twist high, twist low, and what their biological potency is to ask, would I have used those cells in that clinical indication if I had this information beforehand? So we're hoping we can validate further the utility of the scale using actual clinical isolates that were used in patients. And so I'll end there. Just acknowledge people in my lab. So Sid is a staff scientist in my lab. He's been with me for quite a while. He's done pretty much all of the work on the clip scale. Chris Hager is another staff scientist. He does our bioinformatics. And Corey's a grad student who's also working on this project. She's actually doing a global chip-seq to find out what all the twist targets are in our MSCs, which then we hope we can use that to add new metrics to the clip scale. And thank Dr. Ortiz at Pittsburgh, a longtime collaborator, who basically has worked with us to do all the lung injury studies. So thank you. I'll take any questions.
We learned a lot about MSCs, I think, in the previous trial. So, and there's a responder, perhaps responder, non-responder elements maybe there. So, do you think that this is a question, in your opinion, so there's no data, IPS cell lines, donors, and even ES cells, do you think they'll have this kind of a responder, non-responder, the appropriateness in terms of, the what cells, therapy, et cetera? Do you suspect that this could be applied as well to other cells?
Yeah, I think absolutely. The question is, what path of lineage specification do you use to make your ES cell into an MSC? So we've gotten IPS and ES-derived MSCs from a number of different labs, and they don't come close to the robustness of bone marrow-derived MSCs. So the question becomes, what path do you choose and what selection do you use? So do you do a SOX2 positive neural epithelial progenitor? Do you select for PGF receptor expression? How are you driving your ES cell? I think if you get to this twist GLI1 positive skeletal progenitor that we think we work with, then it should have the same properties because twist is playing a predominant role in driving the MSC phenotype in that population. But this is the real issue. I mean, Dan made a comment which is sort of flippant, but we don't know what we're doing. I mean, in a sense, we don't because the bone marrow MSC population is quite heterogeneous. So if you go back and look at lineage tracing studies in mice, everybody's got their favorite marker for what the skeletal stem cell is in bone marrow. And it's actually a heterogeneous population. So twist is really driving neural crest differentiation through EMT into these migratory neural crest cells that then invade the cranial mesoderm and Twist and Gli1 are truly the stem cells that define the dermal and skeletal fates in the cranial mesenchyme. Whether it does that in long bones, we don't even know. What's relevant is you get human MSCs from flat bones, not long bones. So I think this is quite relevant. What's interesting, in mice, we all study MSCs from long bones. So we do a lot of work in our lab with looking at different species-specific differences between mouse and human, and they're quite significant. So it turns out in mice, Twist2 drives all of these phenomena. If you silence Twist1 in primary mouse bone marrow MSCs, it doesn't do anything. It's all Twist2. But in humans, Twist2 doesn't do anything if you silence Twist1. Now there's an important interplay between Twist1 and 2, so they're interrelated. But so, you know, to me, I don't like working with ES and IPS-derived MSCs because I'm not sure that they recapitulate the biology of the bone marrow-derived cell. And, you know, when people send me those cells, I want to know exactly how did you fate them and what do you think you have in the end. So if you can drive them toward a cranial neural crest progenitor that, you know, expresses twist and snail and these things, then yeah, I think this will hold up because they should be equivalent cells.
So take that a step further, because there's a push, as you know, to utilize IPS-derived MSCs, particularly from a manufacturing standpoint, because you can generate trillions of them, right? But your point about the GC-rich and the transcriptional regulation and the epigenetic regulation is critical, I think.
Yeah, so
IPS-derived MSCs still have presumably some sort of epigenetic memory of their origin.
yeah. Well, and I don't think anybody, I mean, I don't follow the IPS field closely. I don't know what the role of twist is in any of that, and if people even look at it. So you're right. I think we're going to find that epigenetics plays a really prominent role here, and so then when you do reprogramming, you know, are you retaining the level of regulation at the twist locus that you need? Because at least for these cells, twist is a very dominant player in their phenotype. Lots of questions.
What we've done in our lab is we treat the MSCs with interferon or TNF to make them more immune modulatory. Does that affect twist levels?
Yeah, so interferon downregulates twist. Interferon gamma directly downregulates
That would make
twist.
sense.
Yeah. The problem with interferon gamma is it's a nuclear bomb. I mean, it shuts off ribosomal protein synthesis. And it's interesting because twist regulates, some of the direct twist targets are ribosomal proteins. And so when you knock down twist, you see some downregulation of some ribosomal proteins, which are consistent with when you hit the cells with interferon gamma. So they share some of these. So some of the effects of interferon gamma on ribosomal protein synthesis, I think, is mediated by twist, because twist is regulating some of these genes. But interferon gamma is, I mean, in our hands, we treat cells with interferon gamma. They just...
What about TNF?
We haven't done much with TNF, to be honest with you. So
there's I guess, a big push, at least in the research, to try different sources of MSCs, whether it's adipose-derived or umbilical-derived. Do you see any differences if you cultured any of those different derived MSCs in terms of twist levels, or is it just like they're all over the place no matter what source?
yeah, so I'm embarrassed to say that we get this question all the time, and we've never done the experiment. So we haven't looked at twist levels in adipose or umbilical cord derived cells. And it's, people send us cells to do it, and we just never get around to doing it. It's something we have to address. If you believe the bone marrow cells are some kind of having tissue parasite-like cell, then it should hold up, because that would be what you would suspect the source of MSCs from adipose to be. But we just haven't done that experiment. We actually really need to do that. We haven't, well, yes, because we haven't really seen, so we'll get donors, we'll grow them out, and before we freeze them, we'll do some analysis, and then we'll go back. We haven't seen any real effect of frozen in terms of this. But, I mean, I certainly tend to agree with Jacques that, you know, if you take cells out of frozen state, they're probably inert for some time until they can recover. I mean, this is one of the complications of clinical therapy. We all use fresh cells in culture and use our animal models. We don't typically thaw a vial and infuse it into a mouse. So I think that's another one of the confounding variables of trying to relate translational studies to clinical outcomes. Any more questions? All right, great, thank you. I guess that concludes our session. Really appreciate you showing up early this morning. And again, I'll apologize for Kat and John for not being able to be here. I think John had a conflict and Kat just couldn't get out of DC with the thunderstorms. Yeah, and our third speaker on the program was actually from Hopkins, Dr. Ling, and so we're assuming that his flight was canceled as well, so we apologize for that. But again, thank you for attending and enjoy the rest of the meeting.